I start with a discussion on complement system. I want to add some information. I had dealt with adjuvants. First part of my lecture, I dealt with adjuvants. At that time, I had mentioned that the Bharat Biotech in Hyderabad is not using the adjuvant for their COVID vaccine, that is Covaxin. Yes, it was true. But same day in the evening, I came to know that since the immune response shown by the vaccine in the phase 1 trial and the phase 2 trial was found to be very poor, they have now adopted a vaccine. They have started using the vaccine for the Covaxin, and this is imported from United States, manufactured by a Kansas-based company, Virovax. And the name of the compound that they are using as an adjuvant is all hydroxy hydroxyquim, QUIM, all hydroxyquim. It is supposed to be a lipid based compound, and they are using that compound as an adjuvant to boost the immune response and try and see whether it will be better than what they had got the response in the phase 1 trial. Okay, let's see now how the response will be and how the vaccine will do. Okay, please note that it will be a different kind of an adjuvant, all hydroxyquim, QUIM 2. It is a compound imported from abroad. So we'll talk now today about the complement system. I had mentioned about this briefly when I dealt with innate immune response. Remember, in the alternate pathway of complement. All right. So alternate pathway of complement is also a part of the complement system. And in the complement system, you can classify the complement into classical complement pathway, alternate complement pathway, and lectin pathway. Okay, lectin pathway. So classical complement pathway consists of about 9 complements. There are 9 complements, C1 to C9. Some of them are glycoproteins and some are protease enzymes. Okay, all these 9 complements, some of them are produced by the macrophage and some are produced by our liver. And these complements are produced only when there is antigen antibody complex in our body. Whenever there is an antigen, the antibody will be produced. It is going to hunt down that antigen and form a complex with that. And I had told you that the complements facilitate removal of the complex formed by the antigen and the antibody. Either it is a preceptin complex or it could be even agglutinated complex. If these two complexes deposit in our body, it will be dangerous for the body. It may deposit in small blood vessels, capillaries, or vital passages of the body because of the excessive accumulation of the antigen antibody complex, and it will block that vital passage, would cause real damage. It may even block the blood vessel. In order to prevent all this danger, the complement system gets activated whenever there is the complex of antigen and antibody to get rid of that antigen antibody complex. Okay, to begin with, actually the complement C1 gets activated whenever there is antigen antibody complex, and C1 consists of, it is a molecule like this, it looks like this. C1 complement complex looks like this. It has a handle that is called a stalk, and it has the whip with head. There are 6 heads there. Okay, 6 heads which are almost like spherical heads attached to that whip, and those heads, in fact, which are there, are the ones which are going to interact with the FC arm of the antibody, that is at the CH2 domain of the antibody, that any one of this head will be attached to this the region CH2 domain of the antibody, and another head will be attached to another antibody. That means there is a necessity of two IgG molecules, immunoglobulin G molecules, sitting side by side at a distance of about 100 angstroms with FAB site of the antibody is engaged by the antigen. That means the antibody has already formed the complex with the antigen, and two antibody molecules sitting side by side. In between that, the complement will go and bind, and it will bind like this. You can make out in this diagram in the pathway, the antibodies are bound there to the microbe, and C1 complement has gone and bound to the FC arm of the antibody. Okay, we'll come back to this. So the C1 arm, moment it goes and binds through its head to the FC region of the antibody, there are two components in it, that is C1R and C1S. Okay, the C1Q component is this major part made up of stalk, and the whip and the head is C1Q. And there are two components, C1R and C1S. And C1R is also split into C1R1, C1R2, and C1S is also split into C1S1 and C1S2. There are two domains of each of these, R1, R2, S1, S2. Okay, and these two domains are proteases. They are proteases. They will get activated. Okay, they will get activated when the C1Q with its head engages with the FC arm of the antibody. Once these proteases are activated, the two complements get attracted to them. They are proteases. Okay, the two complements get attracted to them, that is C2 and C4. Okay, remember I've told you there are C1 to C9 complement, and each time one complement component may act as an enzyme and the other one will act like a substrate. It is like an enzyme substrate reaction, and it will go in a chain like fashion in a cascading manner. Therefore it is called as complement cascade. Complement cascade. First one to get activated is C1Q with its other components C1R and C1S, and it recognizes the antigen antibody complex, binds to the FC arm of the antigen antibody complex. Okay, and then now it is responsible for reacting with C2 and C4 complement. When it reacts with C2 and C4 complement, that protease action, it will split that C2 and C4 complement into two small parts, of which it will be C2A and C2B, excuse me, C4A and C4B. Remember, always A component, that is 2A or 4A, all this which are formed, they will be smaller in size compared to B component. Excuse me, C4B. C4B is bigger, C2B is bigger. So C2A and C4A that are formed are smaller component. Now what happens? Once the C2 and C4 are split by the protease action, these two components of which C4B remains attached to the bacterial cell wall. C4B remains attached to the bacterial cell wall. Now, let us presume that the antigen is a particulate antigen, which has formed a complex with the antibody. As shown in this diagram, bacterial wall, C4B is bound there. As soon as C4B binds there, the other component that is split, that is C2A, joins it. It will form a complex C4B2A complex. Okay, and other components will be released free, of which C4A has a major role to act as a chemotactic substance. Okay, C4A is going to play a major role as a chemotactic substance, which I will come to that little later. For the time being, you keep in mind now, another complex is formed now from the split components of the C2 and C4, and that split component will associate to form a complex called as C4b2A. And that component which is formed on the bacterial cell membrane is called as C3 convertase. It is now a protease enzyme. C4b2A is a protease enzyme that will now attract C3 complement and split that into C3A and C3B, of which C3A is going to be released, and that C3A will act as anaphylatoxin, once again a chemotactic factor, whereas C3B will join C4b2A complex which had already formed on the bacterial cell membrane. Now you have a complement complex C4b 2a 3b. Is it clear? It will form a complex C4b 2a 3b. So this complex now acts as another enzyme which is called as C5 convertase. C5 convertase. So the classical complement pathway begins with C1 complement. As soon as C1 complement is activated because of the formation of antigen-antibody complex, the two complements are simultaneously split, that is C2 and C4. And when they are split, the C4b complement that is formed will remain with the bacterial cell membrane, and it attracts the C2a complement that is formed by splitting C2 and forms a complex. It will be C4b2a, and it is shown in the diagram here, C2A for C4b, no problem. And then that will attract, it is a C3 convertase enzyme, attracts C3 complement and splits that into C3A and C3b. And that C3A, C3b, of which C3b will remain with C4b, 2A, 3b as a complex, which is the C5 convertase. It is C5 convertase. Okay. Now that C5 convertase will react on the complement C5. When it reacts on the complement C5, it will split that C5 into C5A and C5B. So when it splits that into C5A and C5B, the C5A will be released as anaphylatoxin. It will join. Now you have three different chemotactic factors, that is C3A, C4A, C5A released as chemotactic factors. And now C5B, after it is formed, it is not going to form the complex with these as a separate complex. It will form a complex of its own, and that C5B becomes a foundation. C5B becomes a foundation on the bacterial cell membrane for the buildup of other complements. Here onwards, after the formation of C5b, there will be no more splitting of the complements. Other complements that are going to be attracted once the C5b is formed are not going to be split. Only up to C5 complement, the protease action takes place and complements are split into A and B component. But after C5b, there will be no splitting of the complements. The C5b becomes the foundation. It remains attached to the bacterial cell membrane. Then it attracts C6 first, then comes C7, then comes C8. Once the association of C5b with C6, C7, C8 is formed, it makes an associated complex which attracts C9 complement. When the C9 complement is attracted, it is going to be more than one C9 complement comes and joins it. And therefore, it is poly C9 complex. As shown in the diagram here, the C9 complements tend to about 15 C9 complements come and join there. And they align themselves in a spherical manner with a hole in the middle. They are now assembling on the bacterial cell membrane. Remember that. On the bacterial cell membrane, the C9 complement has aligned itself in such a way that it will create a pore on the bacterial cell membrane, because the other complements had found a foundation there on the bacterial cell membrane. And that pore will be about 100 angstrom in diameter. Okay, and this complex is called as membrane attack complex, MAC. So membrane attack complex, once it is formed on the bacterial cell membrane, please remember, it need not be only one. It could be large number of membrane attack complexes, and it will look like this diagrammatically. And in electron micrograph, the membrane attack complex looks like that. All these are C9 complements, and the C5b, C6, C7, C8 are here. And you see there are pores created on the bacterial cell membrane, which you can make out in the electron micrograph, which looks like a poke mark, as though the poke marks that are seen on a golf ball. So once the pore is created on the bacterial cell membrane, because of the membrane attack complex, the cytoplasmic content of the bacteria will pour out. It will ooze out of the bacteria. And that is how the bacterial cell dies. So the complement facilitates lysis of the bacteria, or the complement facilitates killing of the bacteria. All right. What if it is not a bacteria? It could be a virus, and virus does not have the cytoplasm. Or it could be a soluble antigen. Okay, soluble antigen. And in the beginning of the lecture, I mentioned that complements facilitate removal of antigen-antibody complex. Okay, cellular complex made up of bacteria. Whenever there is agglutination of bacteria, this is the end result of the bacteria. That is, complement is activated in the form of a cascade. I'll come back once again. First, C1 is going to be activated, which has the protease enzymes made up of C1R and C1S. And these protease enzymes are activated because C1q establishes the contact through its head with the FCR of the antibody, which has already formed the complex with the antigen in the FAB region. Two IgG molecules will be required side by side, sitting side by side at a distance of 100 angstroms to activate the complement. Once the C1q is activated, it is the chain reaction which is not going to stop. It is like a fission reaction in the atomic bomb explosion. Okay, so it is kind of a fission reaction only. So C1 will split C2 and C4, and of which C4b remains attached to the bacterial cell membrane, which attracts C2a, and it forms a complex C4b2a complex, which acts like an enzyme, and that enzyme will be responsible for splitting C3 into C3a and C3b, of which C3b will remain with the complex as C4b2a3b, which acts like an enzyme and splits C5, and then the C5b is formed. Okay, once the C5b is formed, rest of the other complements come and bind there to build up the complement to make it as a membrane attack complex. Now, what if the precipitin reaction has taken place and it is not a bacterial cell or any cellular surface? Only precipitate has been formed because of the antigen-antibody complex. As soon as precipitate is formed, once the complement builds up to C4b2a3b complement, all right, it will build up. Why? Because the antibody will attract this complement and it will form a complement here, complex, and then it will build up to this reaction, that is up to C3b. But since the membrane surface is not there. It is not there. It will help in phagocytosis of that antigen antibody complex, phagocytosis of that antigen antibody complex through C3b receptor found on the antigen presenting cells or phagocytes. Neutrophils and macrophages will facilitate removal of that antigen antibody complex. Okay. Otherwise, we have told you that C3b complement can be formed without the presence of antigen antibody complex. Okay. So antigen antibody complex is not absolutely necessary for formation of the C3b. C3 can get activated autonomously whenever it detects the antigen in the body. And that C3 which is built up autonomously, that is capable of starting the alternate pathway on its own as shown in this diagram in the right hand side. And that C3b which is auto split from the C3 is going to form a complex with that preceptin complex and facilitates its phagocytosis through the C3b receptor. Similarly, whenever there is a viral particle in the body, that viral particle being an antigen, it will stimulate the alternate pathway of complement, and that alternate pathway of complement will undergo activation, and C3b is formed because of the auto splitting, and that C3b deposits on the viral surface, and that viral surface is going to be now phagocytized by the antigen presenting cells or the macrophages and neutrophils. Even then, if the virus has succeeded in production of the antibody in a person, that antibody will form a complex with that virus. And as soon as the antigen antibody complex is formed with the virus, the C3b can be activated autonomously, and that can come and bind to the Fc region of the antibody and facilitate uptake of the antibody. Okay. In addition to that, only when antigen antibody complex is also formed without the involvement of complement also. I had mentioned that neutrophils have Fc receptor. Macrophages have Fc receptor. The Fc receptor of the antibody can be lodged, Fc receptor of the antibody can be lodged on the macrophages or the neutrophils, and antigen bound here in the form of a virus or precipitate is also going to be endocytosed or phagocytosed and cleared. So different ways of clearing of the antigen antibody complexes are there, but the complement is the major thing that facilitates removal of the particulate antigen. Please remember that the particulate antigen in the form of cell is going to be removed with the help of classical complement pathway, and that facilitates removal through the activation of complement cascade and activation of different complements from C1 to C9, and the C9 builds up on the membrane of the bacteria, and that bacteria oozes out its cytoplasmic content and dies. Okay. Then I had mentioned a lectin pathway, which is another pathway which gets fed into the classical complement pathway. Here, all the three complement pathways have been shown. Why? Because they can be fed, even though they have started innately, they can be fed into the classical complement pathway at different levels. C3b, when it is formed from the alternate pathway, it can be fed at that stage where C3 convertase reaction is taking place. Same way, lectin pathway, which is nothing but lectins or a special type of proteins, okay, which recognize mannose sugar complex, okay. So therefore they are called as mannose binding lectins, MBL. It is shown in the diagram here, MBL. MBL stands for mannose binding lectins, and lectins facilitate activation and destruction of the antigen, bacterial antigen, through this pathway. And these lectins behave like antibodies. Okay, they behave like antibodies. They are also lectins are also glycoproteins, but they are not true antibodies. Okay, they are not true antibodies. I'll give you an example. You might have all heard about horse gram. Horse gram. Okay, in Kannada it is called as huruli. In Konkani it is called as kulit. This horse gram, when you soak it in water overnight and next day you grind it, you will get a milky suspension from that ground horse gram. Separate that milky suspension and then you take any person with A blood group, human A blood group, irrespective of A positive, A negative, whatever. And then you add that milky suspension, one drop to the A blood group RBC. You will see RBCs will undergo agglutination. It's a very, very surprising reaction. Means lectins, which are there in the horse gram, acting like an antibody, causing the agglutination of the RBC. Okay. Now, coming to the point where the lectin pathway, which is capable of recognizing the mannose sugar complex on the bacterial surface, gets activated, and it is responsible for the activation of an enzyme, okay, which is called as mannose binding lectin activated serine protease. Okay, the lectin pathway is responsible for the activation of MASP, that is mannose binding lectin associated serine protease, MASP, and that enzyme is responsible for splitting of the proteins on the wall of the bacteria, on the wall of the bacteria, and bring about opsonization of the proteins on the wall of the bacteria. And it resembles the C1q complement reaction. Okay, C1q complement reaction. Therefore, the lectin pathway comes at the same stage where C1q is activated, and it feeds into the same stage alternately when the classical complement pathway is going on, and whatever has happened through the lectin pathway, it will get fed into that pathway, and further destruction of the virus goes on from there, continuation as a membrane attack complex from there onwards as C3b and C5b complements. Okay, so alternate pathway of complement starts independently as an innate response. Lectin pathway of complement starts independently as an innate response against the bacterial mannose carbohydrates. Okay, all get fed into the classical complement pathway, which has already started the reaction. But remember, classical complement pathway comes only after the acquired immune response has started. It is not innate. Classical complement pathway will begin only when acquired immune response has started. That means it is more active after the antibody production. And I told you that seventh day onwards after the infection, the antibody titer starts increasing. And when the antibody titer starts increasing in the serum of a person, there will be more encountering of the antigens that have entered the body, and then the antigen antibody complex starts building up in the 7th to 15th day of infection, and that the building up of the complex will initiate the production of classical complements from the liver as well as by the activated macrophages, and it starts getting poured into the serum to get rid of that complement, the get rid of the antigen antibody complex. Okay, so it is how the classical complement pathway comes in the acquired immune response to get rid of the complex of the antigen antibody. But alternate pathway, I had already mentioned to you that it doesn't wait for the classical complement pathway to start. When it has already formed the C3b complex, and the complex that is developed like this C3bb, which we had discussed in the innate immune response, which will build on the surface of the bacteria and attract a stabilizer protein called as properdin and that will form the complex on the surface of the bacteria, which will facilitate the bacteria to be phagocytized through this receptor of the C3b or endocytosed and destroy that bacteria through the phagocytosis or endocytosis. Meantime, when this complex has built up on the bacteria, it has caused the denaturation of the bacterial cell wall, and I use the word opsonization. Okay, and I will use the same word opsonization when the lectin pathway also starts. When the lectin pathway takes place, also the lectin pathway, because of the binding of the lectins to the mannose sugars of the bacteria and activation of the mannose binding lectin serine proteases, it causes the opsonization of the bacterial cell wall. Okay, and that also is responsible for the destruction of the bacteria. So, two independent pathways which happen as an innate response in the complement pathway also help in destruction of the bacteria, also help in destruction of the virus, that is only through the alternate pathway, not by the lectin pathway, because mannose sugars are not there in the virus. Alternate pathway only facilitate destruction of the virus by endocytosis or phagocytosis. Okay, and the classical complement pathway comes late, and that is after the acquired immune response has begun. Now, remaining other consequences of the classical complement pathway. What are the consequences of the classical complement pathway? Now you realize that when the infection has taken place, there is a series of reaction that is going to take place by the activation of these complements, and it is going to cause a severe inflammation. And that is what is experienced by the people who are infected by the coronavirus. Okay, the coronavirus also initiates this pathway. Fine. So one thing that I told you is opsonization. Opsonization is the word used only for the cells. Okay, the membrane destruction by the formation or buildup of the complement on the membrane of the bacteria is called as opsonization. I already explained to you about the membrane opsonization by the classical complement pathway, as well as the lectin pathway, as well as the alternate pathway. Any of these three pathways are capable of bringing about the denaturation of the bacterial cell membrane, and that will result in killing of the bacteria indirectly without lysis. Okay, without lysis, it is as good as starving the bacteria to death. The bacterial membrane gets denatured, it will be unable to pick up any nutrients from outside. It is not able to secrete enzymes into the media, and neither it can uptake the nutrients, so it will starve to death. Second important point about the consequences of complement is removal of immune complexes. The immune complexes could be of particulate nature or could be of precipitin nature. Whatever may be the nature of the immune complex, those complexes will be removed by the endocytic or phagocytic route through the macrophage. And I had told you that macrophages are scavenger cells. They are meant for cleaning up of the system. Whenever there is system getting clogged or whenever there is deposition, all these depositions, unwanted depositions, are going to be removed by the macrophage. Next, other cell that is capable of doing this work is the neutrophil. Neutrophil is not a scavenger. It is capable of removing the immune complexes because it does have a C3b receptor on its surface. It also has Fc receptor on its surface. The antigen-antibody complex binds to the Fc receptor and gets endocytosed, or through the C3b it can get endocytosed. Then complements also help in immune regulation. I told you the classical complement, which starts only after the acquired immune response, they will boost the immune response. In the sense, the B lymphocytes that are responsible for the antibody production will respond to the C3 complement, and their proliferation may increase. They undergo proliferation. At the same time, more and more antigen is going to be processed by the antigen producing cells, that is macrophages, or the dendritic cells, and that will facilitate the boosting of the immune response and destruction of the antigen. More the amount of antigen is processed, more antigen is destroyed. Okay, so that is boosted by the C3 complement. I had mentioned that C5a, C3a, C4a may serve as chemotactic factors. Okay, and one of the function of C5a is to stimulate both T and B cells. T cells are responsible for cell-mediated immune response, B cells for the humoral or antibody production. So C5a facilitates further boosting of the T cell and B cell response. Same way, the C5a also facilitates production of IL-1 by the macrophage. And remember, IL-1 is the warning cytokine for the T cells. So the T cells are kept active continuously as long as there is antigen load in the body. As much as antigen is there, as long as there is antigen, T cells are not going to keep quiet. They are not allowed to sleep. And that is through the indirect mechanisms, that is by using IL-1 to trigger the T cell activation, as well as C5a on its own as the T cell activator, stimulator. I had already mentioned that complement building up in the form of classical complement as a MAC on the surface of the bacteria brings about bacteriolysis, which is called as cytolysis. Okay, and then there are other immune cells which are going to be attracted, and we'll come across this once again when we are going to talk about immune complex mediated hypersensitivity, that is type 3 hypersensitivity. There, when the precipitin complex builds up, C5a, C4a, C3a, that is built up because of the activation of the complement, is going to attract macrophages, neutrophils, and eosinophils to that area. In that area where the precipitin complex has been built up, there more and more amount of complement is acting, and because of the activation of the classical complement pathway, these complements which are released as the split up complements are going to be responsible for the chemotaxis of the macrophage, neutrophil, eosinophil. All these three cells are inflammatory cells. Please remember, all these three cells are inflammatory cells. They are capable of causing severe inflammation in that area. And I had already explained to you in the innate immune response, inflammation facilitates the prevention of spreading of infection to different parts of the body. Inflammation helps in containing or, in fact, enclosing the infection in one place only. That is by making the components to come to that area and attack that component in a greater extent. Of course, inflammation leads to damage. There are cells in the neighborhood are going to be damaged. So the C3b complement is also responsible for the proliferation of the leukocytes in the bone marrow, and they are needed in more numbers. That is, neutrophils and macrophages are needed in more numbers in order to, in fact, attack the antigen, or in order to prevent the infections from spreading in the body. So therefore C3b facilitates that, and this is the action shown by the complement when it is activated. Okay, and remember, the complement that facilitates the removal of this antigen antibody complex also causes bystander lysis. Bystander lysis is nothing but the neighboring cells where the complement reaction is taking place may also suffer damage. There is going to be lysis of the neighboring cells When the bacterial cell is destroyed in the vicinity wherever the complement has been activated, there is a possibility of bystander lysis. And that bystander lysis, okay, will result in damage to the local area. And that opsonization reaction, I can briefly show like this here. The bacterial cell bound by the IgG can be phagocytized through the Fc receptor by the macrophage, or the bacterial cell bound by the C3b can be phagocytized or endocytosed through the C3b receptor, which is referred as CR1. CR1 is the receptor for C3b. Fc receptor is for the antibody. Both these receptors are active on the phagocytic membrane surface, and it is through this mechanism the bacterial cells can be endocytosed or phagocytosed and destroyed. Not only that, they have already undergone destruction because of the complement has been built up on their surface, and that complex can be removed from your body. The clearance of the antigen antibody complex in the form of prescriptin is also going to be facilitated as shown in the diagram here, where the antibody with the prescriptin complex is going to be swallowed by the phagocyte and destroyed. So this is how the complement facilitates removal of the complement complex as well as the antigen antibody complex. And I had already mentioned about the inflammation, and the inflammation results in lysosomal activity, and there is tremendous amount of the components secreted, histamine and serotonin, which are going to increase the blood vessel size and dimension, and therefore the complement is responsible for inflammatory reaction. I'll stop here. Thank you.